Hope this is the first slide I have discussed already. This is what we have discussed in the last classes. Why it is coming? Really, I will upload these slides. I think. Yeah. Then, what is we studied about genomics? What is genomics? We have studied in detail. Then, what is genomic assisted breeding? What is the protocol of genomic assisted breeding? How we studied in detail about what is mapping, how we can map the gene. We having a map now where the particular gene is present, and we can use MAS and we can put into a desirable cultivar, or we can improve the particular variety. If we have everything, then why we have to go for genomic system breeding? Actually, where all your markers and everything it is comes under genomics only. You don't think that the MAS, QTL mapping, all those things are not separate from the genomics. They are the integral part of genomic breeding. Okay, but overall, it's like multi-omics thing. It's a multi-omics. We discussed only one part, like we have sequence and we have designed the markers, and we study the one part of the genomics we studied already. Now we can study other part of the genomic assisted breeding by looking at now we can look overall protocol. For example, we have a genome. First, in the plant breeding, what we had to do? First, you had to check the germplasm. You had to search the genes. For example, you want to develop a nematode resistant variety. What we had to do? We want to develop and we want to improve your PB1121 for nematode resistance. What we had to do? We had to search for the genetic source of genes for this nematode resistance. We had to screen a lot of land races or wild species or cultivable species or anything. Okay, any genotype, well, you have to do screening. Means for that, gene discovery and the genetic resource is the first step. Then after that, we have to understand what is the molecular mechanisms. For example, I have identified Pulerada as nematode resistant variety. Then next step is I have to map the genes for nematode resistance. You can use QTL mapping or you can use association mapping. After the mapping, you discover the gene, okay, it is there in present chromosome. Then you have to know why this particular variety is resistant. Is showing resistance mechanism. You need to understand what is the molecular mechanisms of this resistant variety, why it is showing resistance, and how to study. Then comes the different omics techniques, transcriptomics, proteomics, or another. I don't know any omics will come also. Like nowadays, ionomics also there, metabolomics, epigenomics, and the phenomics. Phenomics is actually another part, but that's why I told it's a multi-omic. Not only this genomic, it's a multi-omic. Then we can understand what are the mechanisms of particular trait. For example, resistance only. Why this particular nematode is variety is resistant for nematode? We can integrate. I have done in my MSc. I have used the transcriptomics to understand why the particular resistant variety is showing resistance to nematode. I got several pathways like several ethylene signaling pathway, jasmonic assisted pathway, and auxin related pathways, like some plant hormonal pathways, suberin synthesis pathways. They are highly enriched in the particular, like they are highly uh, expressed In the Pulerada, by looking at the transcriptome history, we can able to see that expression. These only particular they are highly expressed, so that you can say after nematode infection, there may be activation of some signaling pathways like hormonal signaling pathways, some biosynthesis pathways, some secondarily metabolite biosynthesis pathway. Then the plant will synthesize more hormones, more metabolites, then it will become resistant to nematode. I can develop a story on it. And we can use that information in the plant breeding. That is only the genomics breeding. We will study in detail what is transcriptomics, what is proteomics, what is metabolomics, what is epigenomics, what is phenomics. The main aim is to integrate all the omics techniques to understand the gene and the regulatory elements and the regulatory network of the particular trait and what function it's governing. Then finally, after you got all the pathway and which are the genes which are responsible for it, which is highly expressing, which is low expressing, and the gene which synthesis which metabolite, which protein. If you know paka information, perfect information, okay, that we can edit that gene, we can improve that gene, or we can knock down that gene, knock overexpress that gene based on your requirement. This is how the genomic breeding will look. We will, along with your conventional breeding, we will understand the system biology of the plant by integrating what is the use of phenomics, what is the use of epigenomics, what is the use of transcriptomics, what is the use of proteomics. We will discuss in the coming classes. Okay, like each and every omics, I have separate classes. Don't worry if you're not understood today. Then we discussed in detail about these things. Overall procedure of your genomic state breeding. First, we have a genetic resources. We create the mapping populations. We have create this like RIL, NIL, advanced intercross, F2, double haploids, NIL, it's chromosomal substitution lines. Then we can create a NAM population, magic population, whatever it may be. Then we can create a linkage map and we do QTL mapping, or we can map the gene by GWAS. Another technique is the genomic selection we will discuss. Then we'll identify a marker which is associated with the phenotypes. Then we can create a candidate population. Then we can go for MABB or MARS and genomic selection. Then we can improve our trait. Then we can like we can how this is called as QTL introgression stage. These are developmental phase. Means you are developing the QTLs or you are identifying the genes. Then after identification, you had to implementation stage. Means you have to introgress into the new varieties. Any method, any scheme you can use. We have discussed. This is the overall what we are doing in the genomic assisted breeding. Ultimate aim is to find the gene and its function and how it regulates, because the phenotype is not only governed by a single gene. You can't say it's a regulatory network. This year, the question was asked in 2024, what is gene regulatory networks? There is some hub genes. It's like a hub of some other genes, like it's a network. There is the central gene which coordinate all minor genes, hub genes. They are called as hub genes. Then this is the whole thing is called as gene regulatory network. Mainly, the phenotypes are manifested by the different genome regulatory networks in the organism. The question was asked in 2024. You can see. Okay. To understand the gene regulatory networks, to understand which gene having which function, then how it manifests the phenotype, then integrating into the plant breeding is called as genomic assisted breeding. There is a one paper by Rajiv K. Varshane. I took this image from next generation mapping, like the one paper title is there. I will share that paper in the group. Okay, don't. Then how it takes, what is the flow and what is the timeline for the genomic custard breeding. We have germplasm sets. Then you have to, after getting germplasm, for example, you get some germplasm panel of mung bean. Then you have to discover which trait is needed for your improvement. For example, nematode resistance I want to improve. Which traits are necessary? Normally, nematode infects at the root. For example, if why it is a root thin layer, if I make it as a thick layer, then maybe nematode will not going to infect. Like this, we had to understand the trait. For this trait discovery, we had to understand the regulatory network. For example, high suberin mechanism, high synthesis of secondary metabolites was there. We can knock down that second. This is also one trait, secondary metabolites. For example, when you nematode infected, some ions get more released. You can then you can see that which ions are coming out. Then we can use some mitigating strategy for reducing the ionic concentration. There are so many ways we can. This is what the phase of trait discovery. There are several ways to phenotype. For example, root traits. It's very difficult to study through conventional breeding. We can use high throughput phenotyping like multispectral images, like phenomics techniques. Then, okay, you decided the trait and we phenotyped the trait. Then we had to do gene discovery. First, you create mapping population, then QTL mapping, association mapping. Then we can validate them through several functional genomic tools like RNA-seq or through tailing or RNA silencing. We can do many things. We'll discuss what is the tools are available for functional validation of particular genes. Then you can go for allele mining. You can screen the large number of land races or core sets. Then how we can do allele mining? We have to sequence the entire genome. Then we have to go for gene-based association, like candidate gene association mapping. I told. Then Then finally we can do molecular breeding after having the favorable alleles, favorable genes, we know the accurate trait. Then we can use for marker assisted selection, marker assisted recurrent selection, MABB, genomic selection. Then we can improve the particular plant, I food, like whatever it may be. Then you can sell it to seed industry, food processing, whatever it may be, finally to the consumer. What is the advantage? Normally, in
And
conventional breeding, it will take 10 to 15 years, but in molecular breeding or genomic assisted breeding, it will take only 3 to 5 years. Then, how it is started? How genomics is started actually?
after gene discovery, we will go for allele mining, sir.
No, no,
More
no, it is
alleles,
not like
more
that.
specific
No, no,
alleles.
it is not like that. There are different schemes. You can go directly allele mining. There are different things. Okay, it is not like one after the other. Actually, you can take these are club. You can go with this also. You can go this also. It is not like that. After that, it has to be. Okay, it is another strategy to identify different alleles. Okay, then how it
Okay.
started genomics played video. First, when entire our genomics started, when actually the genome sequence, the first crop genome sequence was rice. First plant is
Arabidopsis.
Arabidopsis. Okay, then after the Arabidopsis gene discovery, it's small genome, but they are not cultivated variety. They are not crop actually. Arabidopsis is not a crop. But first crop genome is first sequenced fully saturated genome available is rice genome. The you have to see in detail about rice genome project, when it is came and which paper it's published, what are the things they have discovered. Rice genome projects and different genome projects like rice genome project, wheat genome, what is the reference genome for the wheat. There are different genomics like what is the maize genome, reference genome for the which reference genome is used for sequence. Like that you have to study in detail about all the things. But as a project, rice genome project is very important and human genome project also very important. Entire genomics started when human genome project was taken in 1991 or 1992. It's completed near 2000. Fully complete saturated genome available in 2003. What are the characters, like what is the information we got from human genome projects? There are several information we got. At least you remember several points. Okay. As of now, we see only rice genome project. Actually, you can see total number of the statistics of the rice genome. You can see here, like how many number of gaps are there, how many MBs is there, or like it is a chromosome 1 having 0.33 million base pair, chromosome 2 having 0.1 base pair, like nearly 370 MB million base pairs were there here. Because several, this is the first one, and the reference genome was Japonic cultivar Nippon Bare. Among the domesticated cereals, it is the smallest genome. Okay, it is nearly 432 MB or 380 MB, whatever it may be. It's in the range. Why this range is available? Because whenever they sequence different rice genome, like rice variety, they definitely have some difference in their genome size. They're nearly same, but they were definitely different for several base pairs. That's why sometimes, but all are correct. Don't think that only 380 is wrong, 432, but this range is correct. Okay, if you are, because so many students were asking, like in so many exams, they give multiple options like 380, 430, this one. They given both A and B are correct. But some people have answered only particular answers. It is present in this paper, it's present in that book. It may be right. Why they have given A and B? Because it's a present, it's a range. Then, okay, so what is the landmark varieties which are developed through molecular breeding, especially in the rice? I am discussing only rice. Please go through wheat, maize at least. It's very important. But how to study it? First, you have to check which is the drought tolerant varieties are there, which are the submergence tolerant varieties are there, and what is the major disk. For example, this year they were asked in wheat for the blast resistance. Normally, blast is more predominant in rice, but unfortunately, the blast epidemics was happened in Bangladesh. Luckily, there was a source called as Agilops ventricosa. It having a genes for blast resistance. Then they had to introgression breeding by using Agilops ventricosa. Then they developed blast resistant varieties. It is there, like the significance of identifying the source of the genes is very, that's the importance. There are first I'm dealing with only rice. For example, you take IR61, IR64, and for submerged tolerant varieties, Varna Samba sub 1, CRA sub 1, Ranjit sub 1. And Swarna sub 1 is very, very important. Many times it asked in the exam. What is sub 1? Why? What is the sub 1 gene? From which source sub 1 gene is identified? From, I told in the class now. What is sub 1 gene? From which source the sub 1 gene is identified? Yeah,
FR13A9B.
yeah, exactly. For 13 years. Okay. This is the land race from Northeast India, I think. They have identified sub 1 QTL. Then they had transferred into the mega variety by marker assisted breeding, backrest breeding, mega variety called as Swarna. Earlier Swarna is submergence susceptible. Means after integration of sub 1 gene in the background of Swarna, they have developed Swarna sub 1. This rice variety is can able to sustain submergence for 14 days. Even though rice is completely submerged 14 days, the yield will not be affected. That is the power of single gene. There is another gene called as PUP1. How many of you know about PUP1?
Popcorn 2PL.
Yeah, PUP1 means low phosphorus tolerance. Low phosphorus tolerance in rice. So mainly gone by PUP1 QTL. It also identified in which variety? Kasalna.
Asarat.
Sorry, Kasalat. Then it is transferred into the several background of IR64. There are several mega varieties in the different mega varieties background transferred. Then the rice varieties becoming low phosphorus tolerance. varieties. Okay. Then another QTL called as saltal.
Sir, this is Kasathar, sir.
Actually, that
Kasarat.
is kasal nut. I'm not getting the spelling. Okay, kasal nut something. It will start with KS. Okay,
Okay,
kasal nut or kasalat. Forgetting the word actually. Exact spelling I'm forgetting. But kasal nut. If you find this option, you can just open. Then another QTL is called as saltal one from Pokkali areas. These are very important. This year they have asked about which is the land race of the wheat, which are very important for biscuit making. Naphal, Naphal series of the wheat. I think it's a land race or a species which are important for the biscuit making quality. Then they asked about Sagilops ventricosa, I told, which is for the blast resistance. And they had transferred through genomic assisted breeding. And important QTLs identified also very important, different crops. As we have discussed in rice for phosphorus tolerance, salt tolerance, and for the submergence, very important. And in ASRB, they have directly asked questions from the paper itself. Then another one is called as Pusa Basmati 979 and Pusa Basmati 985. So very, very important. And this year also there was a question asked on this slide. I told all the things, but like how students will perceive or while reading also. We will go for previous year questions, which are the topics appeared in the previous year questions. We will read that topics only. We will forget all the other topics. We'll more focusing on that topics only. We're going for the exam. But if the questions come from the other topics, we will say that it's from out of syllabus. It's not the way. Okay, should read everything so that in a comprehensive way. I'm not telling that you go in detail about all the concepts. At least you should have a brief idea about every topic. Why
But
I
for
told this thing? Yeah,
wheat and maize, sir, there are lot of questions as you said. Which materials
yeah.
or any source can we see, sir, regarding entire
See, I am working in
ingredient?
rice. If any questions coming, like I worked in rice, now I am working in mung bean. If there are any questions related to mung bean and rice, at least I can able to predict and answer. But even for me also, uh, after seeing the paper like wheat and me, see who will study. Even though students are working on it, also the only scientists who working on. See, some scientists are working on some particular trait. They have collected all the review of literature for different project or different paper writing, so that they only know those things. Okay, but it's luck. Okay, don't worry. Sometimes it's a luck. But you have to study crop breeding book for this.
Yes, sir.
And you have to keep in touch with recent trendings. Okay, and landmark papers. Okay, landmark research papers. Many things I have shared. I not shared it, but I will share but I will share near when the exam will near. And I have to create a mock test tomorrow. I will give the mock test of entire molecular breeding. Already created. It's in Telegram. Okay. Then why has taken this specific example means everything is appeared already in the exam. The sub ones, the importance of sub one gene and where the land races, from which land race it has taken, came in the exam. Okay. In ASRB, they are compulsory questions. Then, especially, these are the varieties which released, I think, 2022, I think. Pusa Basmati 1979 and Pusa Basmati 85. This year, the question was asked, that is, the Robin mutant. Robin mutant belongs to which crop? Belongs to rice. I think people from TNI should tell this answer. Because the great scientist had, I think, died last year only. I don't know. It's a very important mutant Everyone is using actually this mutant. Anybody from TNAU? No, no. Tamil Nadu?
Sir,
Yeah. You know about rice robin mutant?
sir, one day you told about him, sir.
Yeah. This is the robin mutant. This asked in 2024 exam. It's related to this example. 1978 and 1985, the varieties which are the world's first herbicide transgene free herbicide tolerant varieties. The world's first herbicide transgene free herbicide tolerant varieties. There is a one particular gene called as AHAS. Means this AHAS gene is responsible for, actually it is this particular gene is inhibited by the herbicide called as imidazopyr. Okay, this is one of the prominent herbicides regularly used, but it is a non-selective. If it is spray, everything is killed. But if you mutate this particular alleles, AHAS alleles, then the plant will become resistant to this herbicide imidazopyr. Then this mutant is identified in the robin. This particular AHS mutant alleles, which are identified in the robin. And actually, scientist name also Robin, I think. Full name I forgotten. That's why he kept the mutant name Robin. And by this as a donor parent, we use this Robin as a donor parent. We transfer this mutant allele in the background of Pusa Basmati 11 to 1 or several Pusa Basmati background, and we develop Pusa Basmati 1979 and Pusa Basmati 1985. This asked in the exam. Okay, please read about this. And who developed it? Dr. A.K. Singh. And our rice team has developed these lines. Okay. Then there are several institutes. Institutes also they were asked. The major, the institute which are working for the rice is IIRR, NRRI, IIRI New Delhi. You should remember all the things. And now what I have discussed in rice now, in the same way, see, XA5, XA13, XA21, they have discovered for bacterial blood. It asked in the ASR. And Saltal also asked in the ASR. And is from Pokkali. Like that, and DRR58 is a both salinity tolerant and bacterial blight resistant variety. And which is the important variety for salinity tolerance in wheat? I also forgotten. Okay. If anybody knows, you can share. Drought tolerant variety in wheat. The soft wheat. Actually, soft wheat, maybe you can expect questions. Because last year in IRA only the varieties developed for soft wheat. In India, normally the wheats are hard wheats. But for bakery products, we need a soft wheat. I mean, the flour should be so soft so that it can able for baking. It easy for baking. Normally, we import the soft wheat from other countries. That's why there are program has taken place to develop the soft wheat varieties. And I forgotten the varietal names. Okay, I have to see. They have released last year, I think last year in 2022 or 2021. Please look at that varietal names. Maybe the question may appear. And which are the important land races in wheat? This year they were asked about. And important land races in, there is one important land race is called as Sikkim primitive. This asked in the 2023. Sikkim primitive. It's a land race of which crop?
Maize,
Yeah. It's a land race of maize. It's known for its proliferance. Means more number of crops per plant. Prolificacy. Okay. And it's mainly like you can use the trait as for baby corn development. There are several things that happen. There is another variety called as toxics. It also asked in the exam. It is a main source of applied inducer. We'll discuss what is applied inducer, maternal haploid inducer, paternal haploid inducer, okay? And there are several things are there. Any doubts regarding today's class? I'm ending the class. This is the only introductory class for the next coming classes. We will discussing about first tomorrow's class will be I think sequencing, first generation sequencing. Why there are different generations of sequencing? Can anybody tell? Yesterday they were asking some questions. That's why I left 10 minutes. Okay. Please ask the questions. We have another 5 to 10 minutes. Somebody texted me, I think Prathamesh or something.
So polycross and gene pyramiding, what difference?
Yeah. What is the definition of polycross? Why we do polycrossing in normal conventional breeding? Forget about gene pyramid. Came later.
The different ingredients like A into B into C into D, like that we cross to incorporate all genes
After
into
that,
a
what we will going to do? After why we need polycross? Where it is used?
In fodder
Order,
crop.
what's order?
After polycrossing, why we need to polycrossing? It's majorly in cross pollinated. The variety is developed through polycross, like we can have multiple crossing. Means we are allowing individual to cross in a population. Then we will maintaining in the isolation. You have to maintain this entire population in isolation. Okay. But gene pyramiding, we can cross multiple individuals. It is the idea is just it's a back crossing, no? Ultimately, it's a back cross. We're having multiple donors. Here, the sharing of the genome from 50-50 chances. It's not only specific gene is transferred here. When the plant is combined with, for example, this plant is combined with any other plant with multiple parents, it can transfer 50% of its genome. Then we are allowing multiple polycross and we are maintaining in the isolation. This is entirely different. In gene pyramiding, we just remember there are different donor parents. By using the markers, we are choosing some particular genes and bringing in a single genetic background. Don't mix up the things, okay?
Sir, then in
Understood what is
polycross.
polycross?
Yes.
What?
Sir, then in polycross, our aim means one of the aim also like to reduce the inbreeding depression also.
Yeah, polycross maybe because normally it is used in cross-pollinated like fudders. Fudders are highly cross-pollinated and they are heterogeneous in nature many times. Okay, like
Okay.
you take pearl millet, it is main that pearl millet to develop pearl millet hybrids, like pearl millet varieties, we are using polycross methods. And polycross is nothing but just crossing with multiple. Then why we are doing this? To maintain the heterogeneity, and they will again we will maintain this entire population in isolation. But gene
Okay.
pyramiding is you are specifically transferring desirable gene from the donor parent. Here the chance of sharing is 50-50%. We don't have any control here in polycross. The genome is shared randomly. But here we have complete control on which gene is going to share on the genome. It's a backcross method, this is a polycross method. You think this multiple backcross to a single recurrent parent. Gene pyramiding is nothing but multiple backcrossing to single gene parent, single background. Polycross is a different thing. Don't confuse it, okay? Any other thing you want to ask? Somebody is asking he not attended the class, so that I don't want to discuss the doubt. Not came for that class.
I only asked
And
this
those, hello?
Abhishek.
Yeah, yeah.
Abhishek. Oh,
No, no, no. You asked that doubt is clear. Another doubt asked by I think Savik, he not came. So that when tomorrow we'll discuss. It's about something. Then any doubts? Like you can ask the doubts in class only. Okay. difficult to text in the WhatsApp because somebody has some different topics itself. It's explaining in the text, it's very difficult. Those who are watching recording, you note down all the points and try to come in the next class live. Okay, so that you can put all the your question in the live, so that everyone can learn. Those who are not there, okay. And these are the not for you guys, okay. You are no, we are attending daily and we are asking daily. No, it's not problem for you. But those who are attending recording classes, actually they are watching recording. Later they list out the doubts and sending in the WhatsApp. It's very difficult to answer. Okay. For listening recording, you may list out all the points, then come to the live class and ask. Okay? You can ask in the WhatsApp for simple questions. Either you can paste the question in WhatsApp, but I will answer in the class. Because so that everyone can learn. See, this doubt we asked so that at least somebody get insights about this topic. And one thing, those who are getting doubts, actually they are at least trying to understand the concept. They're actually known something. When the doubts will arise, if you understood some topics or if you know something, then only doubts will arise. You should be happy for that. Okay. Don't hesitate to ask the doubts. Then any other thing you need to ask, you can ask. No doubts means shall I close?
So best is also to get wheat genomics and rice
Yeah,
genomics.
yeah, definitely, definitely. The spring wheat is actually the reference genome for wheat, and in maize, what is the maize? B73. Can anybody heard about B73?
Yes, sir. It's a famous inbred line, sir.
Yeah, it's a famous inbred line. We discuss in heterologous breeding. I will discuss in detail how actually B73 is developed. And B73, they were asked so many times in the SRB and ASR, ASRB exam. What is B73? It's a reference genome used as in maize. It's a famous reference genome. Okay, the first time B73 only it is completely sequenced. And still now people are using B73 only as a reference genome. In a rice, Nippon burr. In wheat, it's a spring wheat. In a mung bean, it's like a VC 137A something is there. And in chickpea, something is there. And it asked in 2022. In a match the following, you can check. In pigeon pea, can anybody have an idea about what is pigeon, reference genome for pigeon pea? I
Asha.
told Asha, exactly.
Asha.
Yeah, Asha. And in the genome organization class, I already told. Please, at least you should remember some crop reference genome, genome size. It's very must and should. Like either they may question will appear in match the following. Sometimes if you are reading about that, you will get another thing. So what are the advantage? Now maybe there will be pangenomics questions also coming. What is pangenomics? What is dispensive genes? What is hub genes? There are several things are there. We'll discuss. Okay. Don't worry if you don't know these things because you have another separate course called a genomics in plant breeding in PhD. Entire you will deal these things only entire 6 months. That's why don't worry if you don't know these things also. But at least you should know something is there. Okay. Bye.
Sir,
Bye. Good night.
QTLC and all you will teach us, sir, for us.
Yeah, already I made a video you can watch in YouTube.
Yeah, okay. Okay.
First video QTLC only, na? QTLC. I didn't BSA, na? Same BSA, it's the same QTLC. See, by utilizing the principles of bulk segregant analysis, here bulk we screen with SSR mark. Instead of screening with SSR markers, you just sequence the bulks. Then use it for calculation of SNP index. Then you identify the QTLs. That is only BSA. You watch my video, you will going to get.
Okay.
First time Tagaki was discovered. I will discuss transcriptomics in detail. I think I will discuss 2 to 3 class about transcriptomics. Don't worry. Okay. Okay. Bye everyone.
Yes,
Thank
sir.
you.